meadow in this area. So we're looking at a meadow. At the front of the meadow, in this area here, we can see that it's shaded, and that's because behind the lens there's a woodland. And then in this area there is a lot of sunlight, so less sunlight, more sunlight at this particular time of day. And we need to know how to study an ecosystem, so I thought this would be a good example because it's relatively straightforward. It's a continuous meadow, shady, sunny, but also the other difference is that here is relatively flat, and here it starts to go up a hill. So there are two ways that we can study this, two major ways. One is that we can take a big sample of this area, and we can call that, for example, say that this is a 10 meter by 10 meter, and this thing here is called a quadrat. We'll write it down at the bottom here, quadrat. And what we can do is we can sample within this quadrat, and we can do that by dividing the quadrat, at the moment imaginary, but if you were going to do this for real, then you'd have tape measures with you, and you would lay out the tape measures along the x-axis and the y-axis. And so here we're dividing this into a grid, and we would divide it into a 10 by 10 meter grid. I'm sure that this isn't, it's not a 10 by 10, but for argument's sake, it's 10 by 10 meter grid. And then what we need to do is use random numbers for this grid, and we use the x as a x-axis for one set of coordinates, and then we use the y-axis as another set of coordinates. And you can use random number generators. I'll put it up in the sky. Random number generators. These are available on your calculator, but you can get grids of them where you work in pairs and you have your partner select a random number of coordinates. And so what you would get is, say your first coordinate is 2, 2. So you go along 2 and up 2, and then this is the quadrat that you're going to sample. Then say your next set of numbers is 5 and 6. So 5 is here, and then 2, 3, 4, 5, 6, and then this is the next one. And you would keep on going until you've sampled sufficient of this quadrat. Now, that might be 10%, so 10, but it might be a different number, and we'll talk about that in another presentation. Okay, so that way you're going to get a sample of this shaded area, and then you would repeat it in the sunny area, and you would compare those two areas. The quadrat would still have to be 10 by 10 meters, so you're not changing the size. You're still selecting areas to sample within the quadrat by random using your random number generator. And then you're going to compare those two areas. So that's one way of studying it. The other way is to take a transect. And you might remember that this looks suspiciously like a gradient, or an example of zonation, for several reasons. It might be zonation of light. As you come along here, you're getting increased amounts of light, or it might actually be the slope, how steep it is, and that might be related to, for example, the depth of soil, or it could be the soil humidity. Okay, so various things. And then what you'd do is you would systematically, so this is systematic. The other example that we used was, sorry, that's not going to fit in. Systematic was random, but this one, you're going to sample every, 2 meters. And you would obviously describe how often you were sampling. And at every 2 meters, you would lay down your quadrat and you would record in a standardized way what was present in that quadrat. And so now you've got data that is along this line, along this transect, potentially showing you how whatever you've recorded is changing along that line. Okay, so we're going to look next, in the next recording, at how you would, what you could record in your quadrats. The first method for plant sampling that we're going to look at is an actual count. Now you would place your quadrat down and then literally you just count every single, in this case they're yellow kidney vetches on a riverbank, and you would count every single one in that quadrat. Now this technique is only suitable for low density, low number counts. You can't do this for dense populations. And it can be time consuming. You need to be very systematic and very careful. An example on a bigger scale would be counting trees in a quadrat. Say you've got a 100-meter square quadrat. Remember, quadrats can be any size, and you would be able to count the number of trees, either all trees or a specific species of tree. You need to use this method if you're going to look at biodiversity measures, because in biodiversity measures such as the Simpson's Diversity Index, you need actual counts of species. In this case, we've only counted the number of kidney vetch, but for a diversity count, you would count the number of each individual species. And that's your abundance in the equation. Okay, now we're looking at population density for the plants. And what you do in this measure or estimate of plant populations is that you're going to do a fairly similar thing to the actual counts, but this time what's important is that you have the number per area. So we still need to know how many there are, but we need to know how many there are per unit area. So say if this was a 1 meter by 1 meter quadrat, and then we could say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So the density is 4 per meter. Okay, and that's the population density. And then when you're comparing areas, I'm not going to count these, but you can see that there's many more, and so you'd have a much higher density. This is only suitable for plants that grow as discrete individuals. And you can change the scale. So this was a 1 meter quadrat, but you could do a 1 kilometer square quadrat, for example, to look at the number of oak trees in 1 kilometer square. Okay, we're now going to look at how to estimate percentage cover. There are two ways to do this. In your quadrat, you could use what's called the Domin value, which is an estimate. So in this estimate, I would say there are 11 to 25 individuals. And so I would give that the Domin value, and I'll write that down for you. Domin value of 5, and this is a scale of 1 to 10. There's another way to do percentage cover, and that's to use a quadrat with a grid, like we've seen before. I'm not going to draw this particularly accurately, but in that grid, and say the grid is 100 squares, then you would count how many squares. So it's in this square, it's in this square, this square, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 there. So this is in 19 squares, and you might have noticed that one plant can be in more than one square, so it's not the same as frequency or actual counts. So here, if the grid was 100 squares, so 10 by 10, it would be 19%. 19% for the percentage cover of this yellow kidney vetch. The final measure of a plant population that we can do is percentage frequency, and this is very similar to a couple of others that we've done. In this one you need a grid quadrat, and what's going to happen is that again you're going to record the number of plants that are rooted in a quadrat. That would be one way of standardizing it. If they're just touching or overhanging a quadrat square, and so you just need to standardize it, and then you can measure the frequency. And you see I've used a much bigger grid here. So what we've got here is a 1, 2, 3, 4, 5, 6, 7, 8 out of 1, 2, 3, 4, 5, 6, 5 out of 30 squares. And so the percentage cover will be 8 out of 30 multiplied by 100 to turn it into a percentage. And this is another rough estimate. It tells us how common a species is, but it doesn't really give us very much information on how much of the species is present once we've recorded it. Okay, in this presentation I want to show you how different systems can be sampled and studied using just those two basic techniques of transects and quadrats, random sampling or systematic sampling, and it depends upon what question you want to answer or ask for that matter. Okay, so here we've got 6 different systems. We've got a large lake, a woodland. Here we've got a agricultural field. It's grassland, a grass field, but it's also got an unmanaged edge. Here we've got a stream, but notice if you look closely, this area is deeper and also actually faster than this area, which is very, very shallow. Here we've got another meadow or grass field, if you like, but the farmer in this field has left these big, impressive oak trees. And so you could wonder, what's the point of leaving these trees? How much does it impact the diversity? You've got shade here, and you've got sunny area here, and that would change obviously over the course of a day. And then in this area, I wanted to show you this one because this is a study that you'll be able to find on the website. And in this study, we travel from this area here all the way up a hill. And we're going over about 1,000 meters of altitude in that area, over a few kilometers. And so here we've got a change in system from a lowland of about 300 meters to a higher area, which it's not actually 1,000 meters. The top altitude is about 1,000 meters. So you've got about 700 meters of altitudinal change. You can also see here that we've got a lake, the outline of a lake. And you could be asked, how would you study that lake? How would you know what's there? So I'm not going to tell you the answers because that's part of the exercise, but it depends upon what question you're going to ask. So I'll write that here. What question are you asking? Is it about human impact? Is it about diversity? Is it about environmental change? And is it about, let's say, habitat management, which could be linked to the human impact? So all these are questions you could be thinking about, and then you're going to come up with a hypothesis that's linking some kind of biotic factor, such as abundance, diversity in an area, with abiotic factors. And your task will be to choose one of these pictures and to come up with a hypothesis, a question, a hypothesis, and a method for studying them.